Okay, so I welcome you all for today's talk. We have with us Dr. Nikhil Gandasi. He is presently a faculty at Developmental Biology and Genetics Department in the institute. I just talk about little bit his past. So he has done his bachelor's in 2005 at the University of Mysore, followed by his master's in 2007. And then he completed his PhD in 2015 at Subcellular University at Sweden, followed by his postdoc, brief postdoc there. And then 2007, he shifted for his second postdoc at University at Oxford University. Followed by he started a research group at Gothenburg, Sweden in 2019, and then after a couple of years in 2021, he joined the faculty of this institute. His research basically are in diabetes, endocrinology, microscopy. He has a lot of publications, more than 30 publications, and a lot of awards that he got on his way. Ramalinga Swamy Fellow he is right now, and Daniel O'Connor Award, European Foundation Award, Young Investigator Award from Scandinavian Society. Then, I mean, the list is long, so I think I just asked Dr. Nikhil to really talk about this wonderful topic. Microscopes are required in a big way to understand how the biology works. So Nikhil, can you please take over?
Thank you so much for inviting me, Plata, and also a very nice introduction and all the effort that takes in, I understand. So thank you so much. I'm going to speak about subcellular view of islet cells to understand type 2 diabetes. This is an overall, I'll give you an idea of what the biology is about, how we use the microscopes for the questions that we want to answer, and what are the different projects that we're doing, trying to interlink and trying to make you understand. Please interrupt me in between if you don't understand and ask me questions. Yeah? Okay. So diabetes is one of the biggest problems in India. So there are metropolitan cities in India like Mumbai, Hyderabad, etc., where the prevalence of diabetes is very high. You can see 14%, 22% in Hyderabad. You might see here that the prevalence has gone up over the years. It was like 5.8% if you see in 2000, and it has gone up until 2015 to 8.7. Right now it's around 9.1, which is not being documented here, but that's the case. So we were the third largest population with diabetics in India. Now we have reached second. So it can also be first very soon. So something needs to be done, and to understand what needs to be done, what we do, what other people do is what this slide is about. So when you have a meal, there is an increase in blood glucose that happens. So this blood glucose, if it gets too much, then it becomes very toxic. We need blood glucose because we need to satisfy all the organs, right? So when the blood is glucose is very high, there is insulin, there's uptake of glucose in the pancreas, which means insulin is secreted. How many of you know insulin? Okay, good. I'm in the right place. So once the insulin is secreted, that means that it can tackle the blood glucose, which means there is glucose uptake in the other tissues like fat, muscle, liver, brain, whatever you ask. So this means that insulin being secreted means that the glucose is taken up, which means that the blood glucose goes down. So you are very hungry, you eat a meal, there's increase in blood glucose, and then you metabolize it. So all these organs are getting the glucose, they're all happy, they are having energy, which means the blood glucose goes down. What happens in the diabetics? They eat so much, but then there is something called insulin resistance, which means that even though there is insulin being secreted, there is no blood glucose is still very high because the blood glucose can't be taken up by the peripheral tissue. Insulin action is something that's compromised, which is why it's called insulin resistance. The other is islet cell dysfunction. So the pancreas have specialized cells, which I go about. So they secrete insulin. And when you try to overcompensate, when this happens, and you try to overcompensate, then there is something which gets sort of, the cells can't do it anymore. It's like a factory. So you try to put too much pressure that at some point the factory will break down. So that's what will happen, and that's what leads to islet cell dysfunction, which means that insulin is also not secreted. The problem gets bigger over time, right? So most of the work I'm going to speak about is regarding islet cell dysfunction, not so much about insulin resistance. So there's a lot of work in the field done on insulin resistance. I'll not go into those details. I'll just focus on this one. Yeah. So if you look at your pancreas, it's a size of a fist, something like this. And it's just below the bile duct, it's just below the liver. And it has two parts. One is the endocrine part, the other is the exocrine part. So in this, you see green parts there, which is the endocrine part. The endocrine part makes a very small, like less than 5% of the pancreas. But what is pancreas is known for is usually the endocrine part. Because everyone knows that there is endocrine part, which is the islets of Langerhans, which secretes the green ones, which are beta cells, which secrete insulin. And then there are alpha cells which secrete glucagon. When you're fasting, there should be something that keeps your blood glucose high so that you don't die, right? I mean, the brain requires blood glucose. So that is taken care by glucagon. And then somatostatin is one more hormone. I'll tell more about it as we go. Right? So when we look at a beta cell, what happens is when there is a high blood glucose, there are transporters in the beta cells which take up the glucose, metabolize the glucose. You know, glycolysis, Krebs cycle, you've started, perfect. So then there's increase in ATP because of that, as you might know already. So this increase in ATP means that there's closure of KATP channels. So KATP channels exist in very much in the secretory cell. Also in other cells it exists, but more so in this case. So there's a chair there and there's a colleague. So there's one more here. When I was just telling about pancreas, what the pancreas does. So this is regarding the beta cells of the pancreas that it can take up blood glucose. And when there is an increase in ATP flow, there is closure of KATP channels, which means that the calcium channels gain entry. This is critical because these calcium channels can drive these granules which are present there. It looks like these. There are many granules in these beta cells. They can fuse to the membrane to release the content, and the content is insulin. So this has to, the whole process has to happen for the insulin to be secreted, which means that you're alive and you're able to metabolize glucose and things, right? So if you look at a single beta cell, in all of you, there are around 9,000 granules. So there are 2 million, right? And at any given time point, there's only 100 of them which secrete. There are many which are part of the membrane, which we call as docked, around 700 of them, a fraction of them again, or 10% of them. And then there is 10% of these which secrete ultimately. Why so? Why? Because there is something called biphasic response. You don't want it like this, that you eat a meal and you will metabolize everything in one go, right? Again, you will be hungry and again you will be eat. That somehow doesn't make sense. So what body has done is that it takes a meal, there is an increase in blood glucose. It makes sure that it doesn't become toxic. So that's why there is a big response there that you can see, which we call as first phase. Then it's slow response to 1 second. So you can see here the plot, it goes up to 2 hours. So in the 2 hours, you're slowly eating up the glucose, in the sense metabolizing the glucose, which means that you have a satiety, in the sense you are fed well, and then you're still trying to deal with this, right? So we wanted to study like what are the different fractional effects now. So this one, what you see here is in the peripheral blood. If I take blood from you immediately after the meal, that's what you will see here. And you might see here in Type-2s, you don't see any of these. So we wanted to see in the single cells if we can visualize these granules and if we can correlate these to the secretion that we see in the peripheral. Can we link that and this? That was that, right? So we used microscopy techniques. Twice we used confocal microscopy, Dürr microscopy, fluorescence microscopy. How many of you know all of these? Confocal, I think you all know. No? Yes. And then Dürr microscopy? No? Okay. And fluorescence microscopy? Okay. I'll go a little bit into it. So the confocal microscopy is trying to take optical sections of the cells. Okay. So let's say you have a cell here. You try to take sections of these, many sections of these. So you can see only the parts that you want them. But the sections are quite thick though, and you might have a lot of light from elsewhere. So simplest is fluorescence microscopy, where you put on the fluorescent light and everything that is there in the cell, you will be able to measure. This is fluorescence. So most of our events that is happening, like I told you in the previous slide, most of the secretion, it's happening in the membrane. So we want something close to the membrane. So we want to see close to the membrane, which is like this, the part here. So we use Dürr microscopy. In Dürr microscopy, you hit the light at a critical angle onto the glass surface, and then there's something called an evanescent wave. So there's only 100 nanometers that you can see here. This is the 100 nanometers here. So this is what is the turf field, evanescent wave here. So you can only visualize in those 100 nanometers. Very beneficial for us because we can visualize the granules which are close to the membrane. And that is where the action is happening, right? And all the other things, like you can see here, everything is visualized. All the other things is completely black. Gives a very good signal to noise. Very critical for us to get the signal to noise. If you look at the images here, this is a confocal microscope. You can see here that you can see a nucleus of the cell here, and this is the plasma membrane of the cell that you see here. So you can see granules like these here, right? If you see the same thing in the curved microscope, it looks something like this. You can see that. So you're seeing from beneath like this, the cells there. Yeah. So you see those granules there, and you can some of the proteins that are in. Right. So the advantage is that you are just focused on where the action is happening, which is close to the plasma membrane. Yeah. So when you fluorescently label these insulin granules, this is how it looks like in the turf field, like you saw also in the previous slide. So in the lab setup, what we do is in the microscope, we try to put the cells on a cover slip, and then we try to do turf imaging. On top of it, we should stimulate with glucose, because I mean in our body it is stimulated by glucose. So something similar has to happen, right? So we use a patch pipette on top to stimulate them with glucose, right? So trying to mimic what is there in your body, right? So when you do that, what happens is that. So you will see here that there are a large number of granules which try to move around, and some of the granules disappear, as you can see that, right? So there are two, yeah, there's. So it kind of brightens up and then they disappear. So why brightening up and disappearance? Mainly because it's coming close to the membrane and it is opening the content because of these are all acidic in pH. So when they come close to the membrane and outside is neutral pH, right? So when they change in pH means that this GFP, which is fluorescent, pH sensitive, will start to make a flash like this. So you can see where the exocytotic events are happening. And we make very good use of this. So that's why we can visualize single events of exocytosis in the turf. Right? When you look at single events, it looks like this. You can see that they're present there and then they brighten up and then they're lost. So the opposite of this is, let's say you fuse, you release one current, right? So there should be more that is replacing this. It's important. Otherwise, you will at some point have nothing. So that is the problem. So you can see that there's nothing present there, which is like you're seeing here, but there's nothing that you see from signal that's coming from inside. And if something comes from inside and if there's an increase in signal, that's called docking. So you see the recruitment happening to the membrane, right? So this is about the method. So we use this method extensively. So we had access to human beta cells from cadaver donors. So we took those human beta cells, we plated them like I showed you in the previous slide, and we tried to stimulate them using glucose, right? So you can see that there are a few granules which are present there. By the end of the experiment, then you will see some of them underwent exocytosis. So what you can do from this is you can quantify the total number of granules. Let's say you are 30 people here. I'll ask, there are 30 people there. And then I say maybe one or two you did exocytosis. So I plot out what's the exocytosis, what's the density of granules. So each donor, each person, I do this for 20 to 30 cells. And then I try to average it, and that's what makes it solve. So each dot is a donor, and then you will have many cells in each one. So you see a population that's developing here, which is type 2 diabetic. We know that they're clinically diagnosed type 2 diabetic and they have lower density of dot granules and lesser exocytosis that is present, right? So if you go across and so each dot here is a donor, and we try to evaluate it with multiple donors, around 60 to 70 donors. And we again see that the density of dot granules that you see here is fewer in case of type 2 diabetes, whereas in non-diabetic, so there is a decrease that you see there. So this decrease means that there is something that's going wrong with respect to total number of dot granules at the plasma membrane of the cell in case of diabetes, right? So we want to know what's happening here. So we wanted to see if we can first mimic, I was telling you about this blood glucose. If I take glucose from your peripheral blood, this is what you see and think about. But at the single cell, what is the response? Is it in the similar way or not? So to see that, we have looked at single beta cells that we plated, the human beta cells. You can see a very nice biphasic response here. Well, not as good as that way because it's single cells, because they all have to come together for the blood, the insulin to come out of the this thing. So that's why it looks very nice. But when you look at single cells, it's quite noisy. That is a common scenario. What you might want to appreciate is that in the type 2 diabetes, there is very little that's happening. You see almost nothing, right? So I told you that the total number of top granules that are present there, they are the ones which release, right? If you try to plot them, the total number, and you try to come down during the timeline, there is a decrease that happens. So something needs to be depleted so that the exocytosis keeps going, right? So in case of type 2s, there's also already less, which I told you already, but the ones which are there also, they don't secrete anymore. So we want to see what is happening to the ones which are there and not secreting anymore. So one thing that we wanted to do was to see if glucose transport is. So because I told you that glucose is a trigger and all that, right? So Pallavi, one of the students in the lab, is working on the glucose transport. She has found out that in type 2 diabetes, there is lesser expression of glucose transporters. So there is one of the problems that we identified. So she's, of course, trying to do that in many ways, trying to starve the cells and then give those cells with more glucose to see if there is more transporters that come out of the brain. More transporters means that there is more glucose that can be taken. This is for another day. I'm just telling you what is wrong. And glucose transport is one of the critical things, and that seems to have an effect. She's still on it. We'll have to see what all we get out. And why glucose transport? Because it's the first step, right? And then to summarize this part, though, regulated exocytosis requires DOCK granules, and these DOCK granules are depleted in case of type 2 diabetes. And this depletion means that there is less of exocytosis that takes place during diabetes. One of the reasons is probably because of the glucose transporters that exist, right? So to go on, what else is wrong? So I told you about docking, the granules being recruited to the plasma membrane of the cell. So we want to see if something is wrong there, because total number of DOCK granules itself was very less, and that is completely unconnected to the glucose transporter. We want to see what is happening there. So we started studying docking. Again, we used turf with something called PALM. Have you heard of PALM? Okay. So this is like a single molecule technique. So what happens is that when you try to stimulate the cells, let's say you have fluorescent proteins, you try to stimulate, they all bleach at a particular way. This bleaching is a common phenomenon, right? So you're giving photons to it. So then they emit the light, and then they start bleaching in a sort of the electrician curve which goes on, right? So, but then if you try to visualize the molecules, if it's a cluster, it goes like this. And if you try to visualize single molecules, they bleach in single step. So you know that it's a single molecule. So this is what we try to use in the palm. We have very high laser powers, and we try to bleach the molecules, which means that you can visualize the molecules to see what is going on. What you see in red here is a granule, insulin granule. What you see in green is a syntaxin, which is a protein which is present in the membrane, which helps in docking. So if I can say that. Okay. So what you see here is that, so there is granule that is present there. Okay, this granule that's present there, and you see two of the molecules coming from outside, trying to attack it and sort of hold it there. So what is happening is the granule is coming from the membrane. The single molecules which are present in the plasma membrane of the cell, they're going together and they're forming a complex step, which is like this. So which is very critical for the docking to, right? So when we look at the cluster formation as well, you see the granule recruitment, and within a few seconds you form a cluster there. The cluster is formed by many molecules acting together. This is very important because what we did was, this is structure of syntaxin. I'll not go into the details of it. So we try to choke off each domain of syntaxin to see what happens. And when we take off one of the domains, which is called HABC domain, which is the main part which interacts with the granule. So there you see there is very less docking that takes place, right? There's almost no docking that you see. So which means that syntaxin is important. HABC domain of syntaxin is important, right? And so this is only one part of it. Mechanistically, in dockings, we got a target. But then that is only one of the molecules. Syntaxin is only one of the molecules. There are at least 30 molecules which are identified in the synaptic transmission. Not here, but in synaptic transmission, most of the mechanisms are very similar. So we want to see at least the critical ones. Do they exist here? Do they recruit? And we saw that after recruitment of syntaxin during docking, you see the syntaxin going up here. there's monk 18, and then within a few seconds, monk 13, calcium channels, etc. So this monk 13 and calcium channel recruitment, we call it as priming. So there's something which sterilizes it, with something which is stuck there, and then there are more proteins which makes it sort of ready, so that then the release can happen, which is the exocytosis, right? So what is recruited here, we call as priming, and you can see delayed recruitment there. So this delayed recruitment, we wanted to evaluate further as well. So at least what we got to know from this is a docking site is formed. And this docking site, something is wrong in this docking site in the type 2 diabetes. So what happens once the docking site is formed is after all these proteins being recruited, there is some sort of so whenever there's stimulation, there is excitosis. All these proteins which are present there, they disintegrate. Why we are doing this is, let's say there is one docking site being formed. Let's say there's excitosis that takes place. So there can be more granules that can come there and again recruit there and again release. So there's no need of syntaxin to be a sort of stopper, right? So we want to see if that is true in the first place. So at least with many of the proteins here, as you can see, that is not true, because once the excitosis happens here, the syntaxin molecules just diffuse on it. So there is a rate limitation that's happening, right? So that means that we are going in the right path with respect to the recruitment of it, and that there is something which is happening. I'll tell you more about that story. And we also looked at lipids as well, because lipids is very critical for this integrity. You might recruit tons of proteins into this place. Ultimately, the membrane, the plasma membrane and the vesicle membrane is made of lipids. So we want to see if the lipids also are present there or they go off. And we saw that in for case of phosphatidylserine, which is one of the abundant lipids there, they also collapse there. And we have also been looking at Type-2, which I'll tell you as we go on. So Type-2 is something that Aishwarya is working on. So what we see with exocytosis and Type-2 is whenever there is exocytosis that takes place, there is Type-2 that is recruited to this. So you can see that Type-2 recruitment that happens. You probably see some sort of spots coming up here and there. And these are more accumulation of Type-2 at that site that you see there, right? So there is change in the protein, there is change in the lipid. Lipid being more sort of transiently coming there within a few milliseconds. And then there is protein which the clusters disappear. That's something, right? And now she's looking at how this is different. If you give them more Type-2, can more excitosis happen? So we'll see where we go. It's just starting. So that's the probe there. So with this, I mean, I'm making short summaries so that you understand. So I wanted to just bring out the fact that the syntaxin is important for the docking to take place. So once syntaxin is done, then there is more proteins which are recruited to that place, which makes sure that there's a stable docking complex that's formed. And once this is formed, then the other proteins can get recruited so that they all get primed, which is what we call it as. So it's sort of a population that is formed there. And then these primed granules can undergo excitosis, and lipids are also somehow involved in this process, right? Type-2 is being one. We are still doing options, right? So all these, what happens to these in case of type 2 diabetes? Some of the proteins that I brought up, RAP3, RIM, and all these, which are sort of proteins which can assist syntaxin. These are proteins which can assist syntaxin. They have less of recruitment in case of dog diabetes. So the ones which are present there, in case of type 2s, there is less recruitment of them for the dog diabetes. So this was something that was very surprising for us, which means to say that, so a granule is coming in here, they are being recruited there. There are some proteins which are forming a complex. There are some which are not forming a complex. And some of these are here. So with syntaxin, we didn't see that. Or with all the story with syntaxin. But these are proteins which were associated with syntaxin. So we went in a converse manner. We took type 2 diabetic beta cells. So type 2 diabetic beta cells means that there is less dot granules, right? So we took type 2 diabetic beta cells. What we did is try to overexpress one protein after the other. Just as an example, we try to overexpress syntaxin. So when you overexpress syntaxin, there is more dots. So this is the density of dot granules. So this is the total number of dot granules. So when we express syntaxin, which is a factor here, then you see more and more docking happening, which means that something is happening with all these proteins so that the docking is reduced. So you can see the proteins that we tried here. You can see RAB, RIM, and all that, which I showed you in the previous slide, the slide here, that it has gone down. And when we try to increase it, there is more dot granules. So it was satisfying to see that. But syntaxin individually also could increase the dot granules. But that we think is because of Rab and Ring, which we are still working on. So that is one of the things. Conversely, what we did was we had access to around 200 donors and we had access to mRNA-seq of all these donors. So we tried to see what is the expression of these proteins in these people, and then can we correlate this expression to insulin secretion? Let's say I take you and say what is the expression of all these proteins and how much insulin do you secrete? That's what it is. And there's something called HbA1c. HbA1c is how glycated your hemoglobin is. So what happens is, if you try to accumulate too much glucose in the blood, you keep on accumulating. There is glycation end products that forms on your RBCs, which is in the blood, right? So that is, it becomes something like this. Then they call it as elevated HbA1c. If you have any diabetic people at home, they usually say, okay, every 6 months we'll go to the hospital, we'll get the evaluation done. So what is the HbA1c? That's one thing that they look at. Oh, HbA1c is too high. They say that, right? Have you heard of this? Anyone? Yes, very good. So that is what HbA1c. It tells you how diabetic a person is, how much glycated hemoglobin. So we try to correlate this with glycated hemoglobin and expression of these proteins. You can see for some of these proteins like, so you can see RAB, RIM and all that. There's a very nice correlation in this case with the secretion of insulin. There is a very nice inverse correlation with the HbA1c, meaning to say that the more diabetic the person is, the less of these proteins being expressed. And also there is less of insulin secretion that's happening. And we can rescue that by giving them more and more, overexpressing more and more of those protein. This is very artificial, though. You can't give protein to people. It wouldn't go to the right place, but still, trying to give you an analogy. Right. So, to summarize this part, there are many proteins which are present in top finals. And the expression of these proteins is very critical. And the expression is reduced for many of these proteins, which means to say that there is less recruitment of top finals. And that is what you see in case of finals. Right. So we can try to express these proteins to recover the phenotype, which is increased docking, but that is not sufficient because it's a very artificial, right? And there are more proteins, so you can't really overexpress everything. So you need something so that there's more expression of these proteins to rescue it. So it's sort of something that we're working on to see what can overexpress this, how can we control it, and things like that. We've been identifying transcription factors, etc., to see if we can handle of it and this. But we are all that. But it's sort of a feeling for where we're going. So until now, I have told you about the granules coming into the membrane, docking, and then priming, which is recruitment of more proteins, and then there's the synapses. I probably convinced you that there's less docking and less priming during the Type-2 diabetes, right? So, are there any specific treatment targets? So, I told you about so many proteins. At least one protein we want to sort of induce them so that we can recover back sort of the phenotype that we see in case of Type-2 diabetes, right? So, within this protein pellet, so we assist a lot of proteins. The story I told you only few proteins. There are more. One of them was very interesting, which is called EPAC2. So EPAC2, why it was interesting is because there is something called GLP-1 and GLP-1 analogs. So most of the people who have diabetes in your family, they usually take sulfonylureas. There's a big class of drugs. And then they take GLP-1 analogs. And GLP-1 analogs, if in the drug form, you might have heard of Exendin, Anyway, these are some of the proteins that they take. What GLP-1 does is, GLP-1 is something that's secreted in the body. Normally itself, there is some sort of a gut hormone, GLP-1, which is required because if there is too much, once the metabolism is over of the food, then the gut should say it's all done. Wait, don't secrete more insulin. That is what GLP does in fact. But then this GLP can control both positively and negatively. So what people start thinking is, can we control our beta cells, whatever is left over in case of type 2s, so that they can stimulate more and more of secretion. So GLP-1 came into being. But GLP-1 has a very short life, sort of lifetime. It dissolves very soon. So they started making analogs of it so you can inject people with this. Right? So we tested with GLP-1 itself in these donors. So what we see is that there is less number of doctrinals that you see, and when you give EPAC2, there is a GLP-1, then you see an increase in the number of doctrinals. In type 2s also there is a little bit of increase, not so much, but there's a little bit of increase. So this GLP-1 is mediated by a protein called EPAC2. So EPAC2 is a cyclic AMP dependent protein. So that is where we thought, fine, let's make a knockout of this and see what is the phenotype. So we made an EPAC2 knockout. When we did an EPAC2 knockout, the blood glucose levels were very high. So it was almost like a diabetic person. So it was always high, it could never be metabolized, it was always like that. So it was that was the case. And in the normal, you see that the blood glucose level once going up, then it comes down very soon. So that was not the case. So so satisfying. So then we again tried to take islets from cadaver donors. So then we tried to treat them with GLP-1 to see if there can be more and more secretion of insulin that can take place, and then there is more of EPAC that can be present there. More of EPAC means that there is more secretion. That's right. So there is HbA1c, which is on the x-axis here. There is EPAC, which is on the y-axis here. HbA1c is dependent on how diabetic that person is. So we, based on this is almost non-diabetic, this is severely diabetic. So we give a sort of GLP-1 to these people. So there is an immediate jump in the EPAC2 that you see there, which means there's more secretion that's happening of insulin. So as you move along the scale here, you still see here, there's a red one. You can give them GLP-1 analogs, and then there is an increase in the secretion or EPAC2 and eventually secretion that happens. But if the person is severely diabetic, nothing much works. You give them as much GLP-1 as possible, nothing much happens. There's no increase that happens with the EPAC2. So that was new for us. We never expected that. But most often what happens is the drugs which are prescribed, how diabetic you are, people don't think. So if you are severely diabetic also, you will get this. That's what usually happens. So what we wanted to say here was if you're severely diabetic, this class of drugs at least doesn't work. That's what's the idea. What works, I also don't know, but this doesn't work. It's important to know that. The person is having less EPAC2, but still is not diabetic. There are some of them. So you can see here, in fact, which are non-diabetic, and here which are the red ones which are diabetic. So I think you wanted to point out that these ones here, right? The one on the far left. So these are completely non-diabetic. So the white ones there you see are completely non-diabetic. So they respond normally. So normally they respond with EPAC and the GLP-1 and things, and it goes. That's a normal thing. Yes. So that I think was known, but what happens here was not. That was what was the critical. So which means that there's less priming and less secretion that happens, right? So we were going after a large pellet of proteins. I told you about EPAC2, I told you about syntaxin at some point, I think, which is here, and I told you about RIM, RAB, and all that. Why do we go across a pellet of proteins? We go across a pellet of proteins because these are all some way or the other involved in the secretion for that. Right? Normally, what people do is they look at the expression of these proteins. Expression is not sufficient. For the function to happen, they have to be recruited at the right place, which is where the microscopy becomes a very big tool for us, because we can see where they are recruited and if they are recruited at the right place. And we can also assess the function by stimulating. We can also do a functional analysis and see if they are recruited to the right place and if they are doing right. That's how microscopy becomes important. And we assess all of these proteins, although the expression was known, through microscopy to see they do localize or not. Right. So one thing that we always wondered was, we are telling so much about the membrane. So to come to the membrane, these vesicles coming to the membrane, there should be something on it to say, go here, go here, go here, something, right? So then we started thinking, can we categorize the protein based on if they are there on the membrane, if they are there on the vesicle? How can we do this? So we used a very simple tool. We did confocal microscopy of with the same proteins on one end, and then we did turf microscopy in the plus one end. Confocal would tell us the detail of what is there in the middle of the cell, and the turf would tell us idea of what is there in the membrane of the cell. So you see here that this is pre-dock, which is basically cytoplasmic fraction, and this is the membrane fraction once it's docked. So you mean to say that is there in the membrane. So with some of the proteins, you don't see any signal there. Syntaxin 1, it's a membrane protein anyway, so it was expected. But with some of them, you see a very large fraction which is already there in the cytoplasm of the cell. And then just keeps increasing as you go along, right? So this was something that was very interesting for us because we kind of got a ligand for the receptor. So we started looking at this, how the complexes form, how they are recruited to the membrane, what are the proteins involved in things. Right. So we went behind this class of proteins, RABs and RIMs there that I pointed out, that fraction which was there also in the cytoplasm and also at the membrane. So we went behind that fraction. So what we try to do is to visualize them with confocal to see if we can see individual granules which are already localized to the granule with that particular protein. And we could see that with RAB. We could also see that with quantity. And then what we try to do is to see once they come to the membrane, does the signal go up? Does the signal increase in the turf field? Because then we can see that there is an increase means that they're bringing it into the turf and then the signal is going up. And we can see very nicely that with monk and RAP3, there's a signal that's going up. And next what we did was to see that, it's like that. So one of them is RAP27, one of them is RAP3. So there are many RAPs present in this cell. And RAP3 is what we're concentrating on. But we just had some other RAP, which is RAP27, to see they also have a similar function. And there we don't see colocalization, that is what is depicted there. And MONC18 is some protein which is present in priming. I think I told you when initially after recruitment of syntaxin, there are other protein. MONC18 was one of them. So this as a readout, you see very nice localization with RAP3, not with RAP27. So with all this, what we did was RAP3, MONC18 localizes, we just knocked out the MONC18 to see what happens to copy. So when we did that, all the other proteins like RIM1, RIM2, RIM binding protein 2, all of these which are present in docking, they also went down. So there's a knockout, there's the Vajra. You can see that there is a decrease that you see there in the decrease, right? So which means to say that there is one which is present on the vesicle. There is a signal that's present on the vesicle, and you try to take out that signal. Then what happens? So there is very less docking, which means to say that that signal was a critical signal to bring it down, right? So you can also see that the clustering also goes down massively. So this shows very nicely the graph where you don't see almost, so zero line is where there is no docking. So 0% of docking was observed and you're not out completely. So and if there is something, if you try to recover it back, then you see that there is still 40% which is present. So which is very satisfying for us because we found a target where we can say that, okay, if MONC18 could be one of the signals which binds to RAB, and this creates a sort of ligand there to bring it to the membrane so that it can dock at the membrane and then the excitotoxin and all that takes. So, but then what is there at the membrane? We are saying so much about the ligand which is there. What is there? So there is a protein which is very well established called RIM interacting molecule or RIM, RAP3 interacting molecule, which is called RIM. So we use that. And what we did was we found out the structure of RIM and we tried to see where it binds to RAP, which is this one here. In fact, it was already established in the neuronal cell. We tried to mimic this in beta, so that it was very similar for that. And then we tried to take out this helix here, this alpha helix, we just removed that. So the idea behind that is when you try to remove it, there should be less docking, right? Because you're bringing something in from the cytosol of the cell to the membrane. If you try to remove the whole thing where it binds to the membrane, then it should go on. So exactly what we saw, there was a decrease there, as you can see. Also with RIM2 or RIM1, both isoforms, you see a decrease there. And the white is white. So it was satisfying to know that there is a receptor and the receptor. It's a very sticking habit. So, but then all this doesn't make sense if we don't know what is, we know only that the localization of these proteins is not happening the right way in case of Type-2s. But functionally, what is happening? Is there something that's blocked functionally? So then we started going along with it again on the neuronal cells. What we found out was that RIM very nicely binds to the calcium channels here. This is a whole complex here. You can see RIM that's present here, and that's here, and then you see the calcium channels which are nicely binding there. So these two form a complex there, the calcium channels and RIM that is present, right? So then we said, okay, now we know something is happening with the expression, there is decrease. What is the function? Maybe it is because of the calcium. So to go and find out what is happening to calcium channels, we again went back to the human cells and we saw that there was less co-localization of these insulin granules with the calcium channels. You can see a decrease there in case of type 2s that you see there. The red one is the type 2. And then again in the type 2s, you see a less secretion that's happening. And that means that there is because of less polar dilution. So we say, okay, the channels are recruited less in type 2s, but the function is also compromised or not. To see the function, what we did is we used a calcium dial, which means that we can see whenever there is a calcium entry, you see a signal. So normally this is how we look at when you look at there's a 205F, which you see the signal there, which is all where the calcium channels are. You can see those places there. So whenever there is an insulin granule undergoing excitosis, we always saw that there is a calcium plume that's formed. So this is a sort of concentration dependent gradient where it kind of increases over time wherever there is a channel. Right. So this is in case of non-diabetic. In case of type 2 diabetic, what happened was there is a sort of there is no sort of point there is no point dependent range where there is an increase in calcium. So what happens is it's everywhere. So this is where the stimulation is, but it's everywhere. The signal is basically everywhere. So there is no particular localized entry of calcium levels. So what we realized is that there is dock granules which release the insulin pattern, and there is a very tight regulation with the calcium channels. This is regulated by REM. So what that story I told you about REM is this is where it's linked to. And here, in case of type 2 diabetes, these channels are away, and then the granule is away, which means that there's no association at all. That's why you see the delay here. That's why there's no correlation at all. So we kind of got the lower expression, what is happening. We could also get a phenotype for it in terms of calcium. So this was very satisfying for us. So we realized that calcium influx is very important for excitosis, which was already well known in neuronal cells, but we tried to see this in beta cells. And that synchronization is lost in case of type 2 diabetes. And the reduced influx means that there's lower number of calcium channels at the place where the excitosis happens. It's not that overall there's a low decrease in expression, which is not true at all. It is just that at the place where it should be localized, that is not the case. So, yeah, so basically, I told you this. So this part, which is the calcium channel, we also saw that there was a decrease. So now we know multiple things. We know that there is less docking, there's less priming, there's less of calcium channel entry. We also know something about loops. So we know that there are completely different things that is happening in case of insulin granule in a single beta cell. So, but then, is it only limited to insulin granules only? It's so much about beta cells. But then there's a lot that goes on, which means that it's in general, the blood glucose is never actually, even in fasting state, people with diabetes, most people who die out of diabetes are dying when they are fasting, when they're not fed, which is a big problem. When they're fed, they have funny things that happen to them, but they still survive. But they die out of hypoglycemia. So we started thinking, what all happens? What are the different granules we can see in the fungus? And can those granules also act on insulin granules? So in the beta cells itself, apart from the insulin granule, which is depicted here, there's also something called extracellular vesicles. So extracellular vesicles exist in all cell types. So this is a common phenomenon that exists. But here as well, we could see this. And this is the EM showing that they exist. So this is the first time we have been able to visualize them. So thanks to Vidya and you. So she was able to visualize this for us, to say, standardize it for us. And we used a kit to isolate them and then put it on the EM and we could visualize it. And it looks very different to the insulin granules. So these are insulin granules. Right? You can see a core that is present there. This is the insulin. All of this is insulin. In this case, there is no core at all being present. So all this we are working on because we want to know the non-core forming granules, which is NPDs, how do they influence the core forming granules? Do they compete? Do they not compete? Can this have a role? These are the ideas. This is very preliminary for that. So we're just able to visualize this non-core forming granules as well in the microscope. Again, the turf microscope. So you can see that. Yeah, okay. So you can see that they do, you can visualize them, they do move along the same lines. But compared to that, when you stimulate them, they don't do much. But whereas insulin granules, for that matter, when you stimulate them, as I showed you previously, it's the same movement. So there you can see they do undergo excitosis, but these don't undergo excitosis, these undergo excitosis. Why this is the case, what's going on, there are a lot of questions and answers. Priyadharshini is leading this work, we'll probably get to know more as we go on, right? And then why are we looking at this? I told you about insulin granules. These insulin granules are massively decreased in case of type 2 diabetes. This is one of the first things that I told you. Also, the MVBs are decreased in case of type 2 diabetes. This is one of the things that we have found out. Why is this the case? What is happening? What are the proteins? How is it linked to insulin granules? Is this a contributing factor? Can we act on it? You can ask a bunch of questions. If we don't have an answer, that's what we are trying to find. Right? And then we'll see how we go on. But there are also different cell types. I told you about an islet which secretes insulin secreting beta cells, there's alpha cells, there's delta cells, etc. Partha, am I doing with time? Okay, yeah. We have 5 minutes to go. Okay, I'll try to wrap up. So there's alpha cells as well, which secretes glucagon, right? So the first problem with visualizing glucagon granules is that we can't label them at all. So first we developed a label, which was in 2020. So then we could visualize them. There as well, there's a decrease, which means that also in fasting state, the glucagon secretion in case of type 2s is somehow sort of compromised. Not as much as insulin secretion, but to a certain degree it is. What is this doing? We're still evaluating. So then we are also looking at phytochemicals, which is what Saptadeep is doing. Why phytochemicals? Because I told you about insulin granules. What are different things happening with insulin granules? I told you about MVPs. Now I told you about glucagon granules. So phytochemicals for us is a way to see, can we modulate any of these? Can we find compounds which can modulate these? That's what she's trying to find. This is one way to see them. We are seeing the downstream signaling. Can we act on it in a different way? That's that. So we'll probably go there some time. So we have understood what happens with insulin granules. We have understood what happens with alpha cells and glucagon secretion to a certain extent. But what about the other one, delta cells? So this we recently got a hold of. We know that the total number of delta cells. So delta cells are told to be sort of a regulator for insulin and glucagon secretion. Those we found out was decreased in case of type 2 diabetes. Mostly Lakshmi and Neetu were the ones who saw this. So this is very preliminary because we just know the number is decreased. We don't know if it is a cause, it's a consequence, nothing we know. So we just said, since it's very exciting that the number is decreased, so we just went ahead and published this anyway, but we still don't know what's the mechanism. So with a lot of questions and very little answers, I think I will end there. So we want to know what is the architecture of the islet and how hormone secretion is regulated and how this all plays a role in case of type 2 diabetes, so that we can understand the small little organoid called islet in terms of insulin secretion, glucagon secretion, somatostatin secretion, and also how they are all helped in case of survival. So with this, I would like to thank the funding agencies and the lab and people who did this work and Partha to help it. Thank you.
Yeah. I think it's a great talk with a lot of insight that you got in the way biology works in case of diabetes. Do you have any question or anything that you want to really ask? You're curious about to know something? Yes.
Yes.
Like multiple proteins that go down because of diabetes. So do you think like are they somehow connected? Like, is one protein going down because of another one going down?
Yes, that is absolutely the case we think.
So if you can somehow like interconnect it, so can you somehow the point of care kind of
Exactly. So now we think that RAB3 and RIM is the initial link, and from that target we can go downstream. But we don't know with point of care or anything like that. There's no targets, there's no inhibitors for RAB or RIM, or there are no activators for RAB or RIM. That is why we went after from RAB or RIM to EPAC, because there were activators there. So where there are activators, you can do something. Otherwise, it becomes very difficult. We have to design something. So the phytochemicals that is coming into play, or also we are trying to see repurposed drugs and things like that. Those are the ones we can probably target the right place, not much more downstream, but we are not yet there.
Okay, anyone else want to? Nikhil, I have something to ask. I just want to know, do you have any, I mean, you can enlighten us with what is the root cause of diabetes in the first place at single molecule level? Like, do you have something to add to that?
So the root cause of diabetes is mostly genetics people want to say it's all risk genes. So these risk genes are mostly transcription factors. So these transcription factors, interestingly, regulate the expression of many of the proteins that I told you about. But how they regulate this expression and at what level and can this be identified very early on would be very interesting. Molecularly speaking, so at some point of time when we are born, these risk genes play more role. As we age and as we get a lot of lifestyle changes, a lot of climate changes and all that, that is when they change. So it would be very nice to see if these transcription factors act on these proteins at the molecular level and how fast they act and where.
Okay. Also, the knowing the docking mechanism and glucose transport and other factors that you talked about, do we have a solution in sight for type 2 diabetes?
People who have already have so many drugs would say yes. I think there are solutions. There are actually multiple solutions. There are many inhibitors, many activators that are found out. But most of it we see is very much downstream. Or once the disease has kicked in, there are probably solutions that exist. But before other solutions, there is none.
Okay. Then also, do we have knowledge about what is the key protein that is involved in type 2 diabetes?
I'd say one, I think.
There is not even for example, there may not be one, maybe two, three, like something that.
But then I would go back to these gene and transcription factors, then PPARG, SLC, and then TCF7L2, these transcription factors, because they are the first ones where they see a change there. I think I would go back on that. There's a lot of work done on that. That's why we have not touched that. But we want to know now these proteins, how they are acted by on that, which is still a niche.
So you talked about MBB and insulin granules, and they said they all go down. Are they correlated in some sense?
So these MBBs, they are known that they can the misfolded proteins that are there. They can dissolve the misfolded proteins. So they can fuse with the insulin granules. This is an idea. That's our guess. Maybe they can fuse with the insulin granules, and the misfolded insulin that is present there, that can be dissolved, is what we are looking at. Because there is many studies from the past decade which show that in type 2 diabetes, apart from less granules, the granules which have insulin also, there is misfolded insulin that's being done. If we can keep them in good state, at least those can do something. Those also we lose. So can we act on that? Because for MBBs, there are so many activators that exist. Drug delivery, that, so many exist. So we're thinking if we can punch that two things together, then can it modify something? That's where the MBB story comes from.
Oh, nice. I think if there are no more questions, then we can close the session. It was great to know a lot about diabetes. Our knowledge about this is really limited, and great to see the knowledge behind it. Okay, thanks a lot.
Thank you. Thank you.
Yeah, he's done. He's done.